Today, we will be discussing our topic on media preparation and industrial sterilization. Large-scale industrial media are not so easy to make, and equally they are difficult to procure. The cost is really very important in industrial sense, and we cannot depend on the components that we use in the laboratory for making of media. A small lab-scale fermenter is mimicking the way we make media in the lab. But in the operation stage, that is in plant scale where huge bioreactors are in use, lot of media in huge quantity are required. And that is our topic of discussion today. How exactly do we prepare these media? What are the ingredients that we are going to use in the preparation of these kinds of industrial media, which will be followed by the way to sterilize these. Now, in any fermentation process, there are two basic processing steps. The first is upstreaming, the second is downstreaming. In the first case of upstreaming, we procure all the materials required to run a bioreactor where a substrate will be converted into a product through the use of microorganisms. And this upstream processing incorporates the requirement of medium and organism, which are to be selected carefully so that the product can be maximally obtained, followed by the fermentation stage. And in the downstreaming, the product is produced which needs to be purified. This goes to the final packaging of the product. From all these cases, we get some effluent wastes which needs to be treated, which are also the case of downstream processing. Now, we are going to discuss how exactly we are procuring the raw materials required for fermentation. It was Robert Koch who gave us the idea of microbiological medium. What is this medium? This is a food base or a nutritional source that are given to microorganisms when we want to artificially culture them. In case of the lab, we can use smaller flasks as you can see here, and the typical stoichiometric equation would be carbon and energy source plus nitrogen source and other requirements which may include some minerals, trace elements, vitamins or other precursors. All together will give rise to cell biomass, the typical product we are trying to obtain in the industry, gases in the form of carbon dioxide, water and heat. So this is a typical equation which is followed in most places where the organisms are growing to give us a product. And the essential requirements are of element carbon, hydrogen, oxygen, nitrogen, sulfur, phosphorus, magnesium and potassium. Other than these, as have already been mentioned, trace elements are also required. Basically, what do a medium do? It produces biomass. If we cannot supply the food, how will the organisms grow? Next, it produces the right byproduct or product and can give rise to acceptable yield in proper productivity sense. There are various kinds of media. Especially in the laboratory, we can use defined media and dehydrated media. Defined media has all the ingredients specifically mentioned and we know the exact composition. It is expensive. Dehydrated media used in lab is equally in the same fashion, but all the ingredients to make of the medium are combined together and mixed as powder just by addition of water. But in lab, we are using these medium which can be really very expensive. We do not bother when we are in an R&D, but think about the huge amounts of substances that the industry produce. Can they produce so much if they are using this high cost material in the beginning? So, the use of this crude media becomes very important in the industries. Like in case of the lab media, the defined media are not only expensive, they are consistent in composition and quality and they are made of pure compounds. Like you can use defined media even in industries if you want to produce certain drugs like anti-cancer drugs or even other antibiotics. While, say, if we want to produce fuel, we will definitely go for very cheap materials, and they should be highly available to us, like production of biofuel from whey. There should be certain characteristics in choosing a particular material to be used as crude medium source. Some of these are enlisted here, like the cost of the material is very, very important. It should be cheap. Availability should be important in other sense that it should be readily available. And transportation costs again becomes very important because industries always think on the economical sphere. The disposal of waste should become very, very significant here because we should not pollute the environment. Then others like uniformity in quality are required, which is not often made by most of the crude sources. Now we are moving on to see these typical medium ingredients which we will be using in preparing media for the industry. The listed above are carbon source, nitrogen source, minerals and growth factors. Then we have water, very essential, buffers, oxygen or air, inducers and elicitors, precursors, inhibitors and antifoams. First, we will discuss about the carbon source. The most important source not only provides the carbon that is required for the huge skeleton that should be produced in the growing biomass, but is equally an energy source. So, the industries, let us see what are the typical common carbon source that are required. To supply sucrose, we have sugarcane, sugar beet, and both these can be produced to give us molasses. Glucose can be obtained easily from corn sugar, starch, or cellulose. Lactose is obtained from milk, whey, fats from several vegetable oils, starch from the maize, potatoes or other grains, and hydrocarbons from the petroleum fraction. And here itself we can have an overview that most of these components, although do provide the required nutrient but cannot always be used. Like in a country where sugar production is not high, cannot go for utilizing sugar for the industries. So briefly, let us discuss these carbon sources individually. The first one that we are discussing is molasses. Molasses is obtained from beet, sugar beet or sugarcane, and the product is a thick brown colored syrup. What is done is, during the production of sugar from either sugarcane or sugar beet, the extract goes for the crystallization of sugar. The remaining charring and dark colored liquid that is produced is the molasses. So this also contains huge amount of sucrose and can be a very good crude source for use by several yeasts, and the production of ethanol often depends on this molasses. The second one that we are discussing is Hydrol molasses. It is very similar. The only difference is the substrate that is used is not sugarcane or sugar beet, but it is maize or corn. The third in line is malt extract. Now this is a very interesting story here, in the sense malt is produced from barley, and most of the malt and malt extract goes for production of beer and whiskey. Now here what we exactly do is, first barley is soaked, then it is allowed to go for partial germination, followed by kiln drying, that is, it is heated to allow partial germination only and further growth not allowed. This gives rise to malt, and then the malt is taken and an aqueous extract is obtained, which is called as a malt extract. This can be concentrated to give us a chocolate colored liquid, and this is the malt extract often used for preparation of beer and whiskey. Then we have starch and dextrins, very commonly obtained from several starch producing plants like potatoes, cassava, and it can be even other materials which we commonly see in the food grade scale. And the powders of starch and dextrins are easily available and can be very well used in the industries as a cheap crude source. Other carbon sources includes sulfite waste liquor and cellulose. We are very familiar with the word cellulose comes from the plant material, while the sulfite waste liquor can be understood as again coming from plant source but indirectly. Now all of us must have heard of the paper pulping industry. In the paper pulping industry, the plant material is first made into a pulp. Doing this requires use of sulfuric acids, and this water extract that we generate after the pulp has been taken, the remaining liquid is known as the sulfite waste liquor. Huge amount of sulfite waste liquor comes down the paper pulping industry, and this becomes a very common and cheap source for several carbon sources. The other one is whey. The dairy industry produces huge amount of whey while making either cheese or similar products, and that is where we can generate this whey. Now whey is bulky and transportation sometimes becomes difficult, and that is why most often these products are concentrated first, like even the sulfite waste liquor requires some kind of refinement and treatment before it is used in the crude media sense. Other carbon sources includes alkanes and alcohols, begins from methane but may be toxic at times, methanol also may be toxic at times, and ethanol very commonly used to produce vinegar or other acetic acids. Then we can also go for lipids, that is fats and oils, which can be used. These are important because if we want to use little quantity and get more energy, fats and oils are the choice. So they go very well with fed batch fermentations. Then we were moving on to the nitrogen source, the second in category. It can be all in the inorganic form, organic form, or in the crude form. Now, inorganic and organic form most often are used in the laboratory, crude forms generally used in industries. But if required, the inorganic and organic forms of nitrogen are also used in industries because nitrogen is required in lesser amount as compared to carbon. The inorganic salts that we generally use are ammonium sulfate, diammonium hydrogen phosphate, or even ammonia. And the organic forms are several amino acids, proteins, and urea. Now, if some of these amino acids serve as precursor molecules, they must be supplied in the pure form, however expensive they are. Crude forms includes corn steep liquor, yeast extract, peptones, and soymeal. And this is where we are going to discuss in little bit more detail. First is the corn steep liquor under the nitrogen source. Corn, that is maize again, during the steeping process, that is soaking in water, generates some amount of liquid that has these hydrolyzed products of the maize. This is called as corn steep liquor. It is most often used in the production of antibiotic penicillin. Nowadays, it has been replaced by pharma media. This contains huge amount of nitrogen and also has other sources like carbon included. Then we are going to see the peptones. Peptones are commonly used in the laboratory. This is a partial digestion product of higher proteins like can be casein or animal proteins, beef proteins or other such proteins. When they are partially hydrolyzed, they generate a huge array of broken down protein components that includes peptones, dipeptides, tripeptides, and oligopeptides. This can also contain free amino acids. These are the common nitrogen source which are often used. Others to follow are yeast extract and soy meal. The yeast extract is obtained from already used up bakers and brewers' yeast. That is, the baking industry and brewery, when they are no longer using after utilizing the yeasts, they throw it. This is a byproduct and can be very well used by the industries which require yeast extract as the nitrogen source. So, the source is Saccharomyces cerevisiae, the yeast, bakers or brewers, and what we extract after it is known as the yeast extract. Soy meal, as we all know of soya beans, when soya beans are pressed to extract the oil, the remaining part can also be further extracted to give us a powdery compound called as soya meal. This is also rich in proteins and can be very well used. Now, we are about to see some of the other requirements used in the industries. The nitrogen and carbon that has already been discussed are required in larger amounts and most often also supplies some of the trace elements, vitamins or other growth accessories. But if required, some of these can also be supplied in purer form. The vitamins and growth factors includes most of the vitamins, amino acids and other precursors. Minerals are equally important. Growth can even be stopped if minerals are not used in the proper amount. Mostly, cobalt, copper, iron, manganese, molybdenum and zinc requires no addition because other crude material like molasses, corn steep liquor will provide these. But the calcium, magnesium, phosphorus, potassium, these at times requires addition. Water is often required in the industries for a lot of activity and as required, deionized, distilled or even simply pure water of good microbiological quality can be used. It depends on where it is exactly getting used, whether for production of steam or to be actually used in media. Now we are going to see some of the other requirements which industry typically demands because of the regulation required in the process so that the product can be procured. The first among them are precursors. This means the building block. For example, for production of L-serine, we require glycine as the precursor molecule when Corynebacterium glycinophilum is used. Likewise, the common precursor molecule always used is phenyl acetic acid, often used for production of penicillin G while using Penicillium chrysogenum as the organism. Let us see what happens. Basically, this is the structure of penicillin where R can be replaced, and if we replace R, this structure is called as 6-amino penicillinic acid. Most microorganisms, that is the Penicillium species, produce these in several combinations. Now, we want a specific antibiotic, say penicillin G or penicillin X, like that. So, the R should be very, very specific. And this R is what we supply in the form of precursor. If we are giving phenyl acetic acid, then the product becomes benzyl penicillin or penicillin G. So, we can see here R is getting replaced with the phenyl acetic acid precursor. This should be incorporated in the medium. Next are inhibitors. Very interesting to note. Say for example, a specific organism can generate two types of compounds. We want one of them. So we have to inhibit one of the metabolic pathways so that the other can be produced. Now, an example that is taken is of sodium sulfite. Obviously, we will not use a lethal dose. The reaction that is during the production of alcohol by Saccharomyces cerevisiae involves acetaldehyde and NADH2 generating alcohol. Now, if we use the sodium sulfite, what happens? This will react with the carbon dioxide in the medium to form sodium bisulfite. And sodium bisulfite next complexes with these acetaldehyde and removes the acetaldehyde. So, the acetaldehyde is no longer present there to generate alcohol, while we have huge amounts of NADH2 being produced there. Then what happens is the dihydroxyacetone phosphate, this utilizes the NADH2 to give us glyceraldehyde 3-phosphate and so we can get glycerol. So this is a very interesting to note that inhibitors we can use and get glycerol from Saccharomyces cerevisiae, while Saccharomyces cerevisiae is generally known to give ethanol. Then we have inducers and elicitors, and these are very commonly used with most often genetically modified organisms. We need to elicit or bring out the product or induce so that the product is produced, often used in medium. Then the others include cell permeability modifiers, means the cell wall needs to be made more permeable so that the product can be excreted out and facilitates downstreaming. One of the example is use of penicillin in production of L-glutamic acid. Then we have to understand that oxygen or air is equally important in any media. We are dealing with organisms which are very well aerobic. So when we are incorporating the medium in a closed cabinet or a chamber called as bioreactor, we should not forget that huge amount of media will cause oxygen deprivation. And as such, air or oxygen in purified form must be supplied into the bioreactor through some mechanism known as sparges. Addition to all this, during the production in a huge bioreactor, because we are using crude media, huge amount of foam gets generated and floats on the top. This is a severe problem and can be minimized by two ways. One is a mechanical aid, not to be discussed here. The other includes use of anti-foaming agents in the media. Some of these are divided into natural antifoams and some can be synthetic ones. The natural ones are the oils that can be used like the sunflower oil or the soybean oil such in very small quantities. And synthetic antifoams include silicone oils, polyalcohols or alkylated glycols. Now, what exactly is the purpose of using them? They are surface active compounds and generally they destabilize the foam structure when used at low concentrations. We should be careful because we are incorporating them in the media, so they should not be utilized by the organism which are growing there. Now we have seen some of the major components which are required in producing media for the industry, and it is again important to realize how to sterilize them. There are so many components which when heated may get charred, like the sugar containing material, some which may produce viscosity, and likewise some products can even be toxic at higher levels. So sometimes individual sterilization of these ingredients are required, and at times we can also mix them and sterilize at a bulk. So sterilization protocols differ depending upon the ingredients chosen. But basically we can understand that the first culture that we will be using should be contamination free. Then all the equipments, media and air generally mentioned, whether oxygen or air, should be sterilized before introduction into the medium, and then only we can go for culturing. So how exactly do we go about sterilizing all these? Media, the other equipments of the bioreactor can be sterilized mostly by moist heat at 121 degrees C, giving a pressure of 15 pounds per square inch and kept at least for 20 minutes. The time can differ and it will depend on the type of the component required to be sterilized. Then other treatments like radiation, ultrasonic treatment, chemicals, mechanical treatment like even some pressure, gases like ozonation can also be used in different parts of the plant, that is the equipments and other accessory requirements. But media should be sterilized by moist heat, and air that we were speaking of should be sterilized by filtration. Here we can see a chamber wherein moist heat sterilization is possible, and several requirements of the bioreactor, maybe in the huge scale, can be all sterilized at a time in this huge autoclave, as we call it, where the principle of sterilization, that is killing of microorganisms, depends on the use of 121 degrees centigrade temperature at 15 pounds per square inch pressure. But in most cases, what happens is we need to sterilize the medium at once. Everything incorporated into the huge bioreactor and then sterilized in situ, means the media is first incorporated, then steam is incorporated into the chamber and kept for the required time period. This is how sterilization in situ is obtained. And we'll see these are some of the cartridges. Filter cartridges most often required for the sterilization of air that has to be done in the industries. The air should pass through these cartridges and filtered before they can enter the bioreactor, because they should not contaminate the contents of the bioreactor. So a brief protocol of sterilization has been followed. although looks and sounds very simple, it is really difficult to maintain aseptic and sterilization conditions in the industries, and most often the loss in yield of a product is because of difficulty in maintaining aseptic and sterilization conditions. So far, as we have seen the different components required in media, several of the sources have been discussed. It is often understandable that an industry really rests on the manpower as well as automated both ways of serving the nation, that is producing products for the public while really working on several kinds of R&D and laboratory facilities which tell us what kind of crude media and other specific sources for carbon, nitrogen and others can be used, and how a sterilization protocol should be maintained to get the maximum product.